Welcome, everyone. It's our great pleasure to welcome you to today's Meet the DM Drug Developers webinar. And we are very, very fortunate to have Sanofi presenting to us. Very interesting presentation ahead of us, as Sanofi is the first company that will introduce a gene therapy approach to the DM drug development efforts. Before we get started, a little bit about the Myotonic Dystrophy Foundation. We envision a world with treatments and a cure for myotonic dystrophy. The mission of the Myotonic Dystrophy Foundation is community, care, and a cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. As many of you know, we provide a whole host of support and resources, and most of these things are available through our website. So there you can find information regarding toolkits and publications. You find information about our support programs. You find our calendar, which is very important for all the variety of events we organize. And you'll find a digital academy with a whole host of information and recordings of all the events we put on and ultimately make available forever to our community. In this slide, I want to lay the seed for a few upcoming events. There's another Meet the Drug Developer webinar in November where we will hear from PepGen. And then I would like to alert you all to the 2026 MDF regional conferences. So please go to the website, pick the one closest to you, and register. And then before we get started, I would like to thank our community education sponsors. So thank you very much to Avidity Biosciences and Dyne Therapeutics for supporting our communication education webinar series. Thank you. So let's get into it. When we take a look at our drug development pipeline, this is really the ultimate indicator of progress and hope. So not only do we have 20 plus programs focusing on finding therapies for myotonic dystrophy, but we have also 11 in clinical testing phases. And here again, we try to update this on a regular basis. There's another update in the pipeline. So visit us frequently for the latest and most up-to-date information. When we look at clinical trial activities, we can identify 4 broad areas of drug discovery. And they essentially follow the central dogma of biology. So going from left to right, we have the DNA with our gene, including the disease-causing gene for DM1, DMPK, and through a process called transcription, where the gene is transcribed into RNA and processed into mRNA. And because of the extensive repeat, cannot fold properly, but generates now these very toxic and very stable hairpin structures, which act as a trap, as a molecular trap for important regulating proteins for cells in the body, and ultimately causes broad disruption of cellular function. So in this first bucket of activity, we have focused on inhibition of transcription and gene editing strategies. The second category of intervention focuses on the inactivation of toxic RNA. The third bucket aims to compensate for the trap protein, so the proteins that bind to these hairpin structures. And the fourth structure is really to correct downstream cellular function. And when you think about this, the more we shift these activities to the left, so to the causal mechanism, the more specific the treatments are. So, like I said in my introduction, today we are very, very fortunate to have Sanofi here. So, Sanofi is interfering with the aims to inactivate the toxic RNA, and they are the first company going into clinical trials, testing a gene therapy mediated approach. We are also very fortunate to have a very stellar team. So we get really the program leadership here. And first, we have Tara Hastings here. Tara is part of the patient-informed development and health value translation team at Sanofi. And she really connects with the community. Tara has a degree from Columbia and from the University of Virginia, and she really bridges between Sanofi in this case and the community. Before coming to Sanofi, she spent extensive time in the rare disease community. You can see here Michael J. Fox Foundation and Parkinson. But most importantly, Tara also knows what she's talking about, as she's also a patient with a rare disease. And obviously, she's very passionate about and being an advocate for patients and families. Then we'll hear from Dr. Pablo Rendo. He's the global project head for gene therapy in rare diseases. So Dr. Rendo is a physician. He received his medical degree from the University of Buenos Aires in Argentina, and he specialized in hematology. He then made a transition from academia to pharma. And again, he focused on hematology and rare diseases. And before coming to Sanofi, he served as the global clinical lead at Pfizer. And of course, there he was also involved in the clinical development of gene therapies for rare blood disorders. Then we hear from Dr. Isobelle Galeon. She's the clinical research director of rare diseases, and she is responsible for the creation and execution of the global clinical development plan and strategy for the myotonic dystrophy program. She's also a physician scientist with clinical training in internal medicine. And she has, in addition to that, a graduate degree in clinical sciences and expertise in genetic epidemiology, biostatistics, clinical investigation, and public health. And she got that from the University of Colorado. She has worked as a principal investigator in many clinical research projects, ranging from common to more rare diseases, and she has a whole host of experience. Last, we will hear from Dr. Didier Rouy. He is the Associate Global Project Head for Gene Therapy. Didier oversees several gene therapy programs, including rare blood disorders and rare diseases, as part of the genomic medicine unit. He's a trained physician, and he also has a PhD in biochemistry. He got that in Paris. Thereafter, he pursued postdoctoral training at Lawrence Berkeley Laboratories in California. And he also has extensive experience from large and small pharma companies. He started his career in cardiovascular medicine with an early focus on gene therapy and stem cell research, and then specialized on gene therapy and gene editing approaches for rare blood disorders. So he also has extensive experience with clinical trials in this field. So with this said, before we get started and hand it over to our Sanofi friends, you all might remember, please ask questions live using the Q&A function of the Zoom event. And then we will try to answer as many questions afterwards, together with the questions you all have submitted when you registered. So please extend a warm welcome to Sanofi. Thank you so much for joining us and presenting us to that today. And without further ado, the floor is yours.
Thank you, Andy. And hi, everyone. It's so nice to be here. And I know I speak on behalf of the entire Sanofi team in saying that we're really excited to have this opportunity to share more with you today. And also so grateful to the amazing team at Myotonic Dystrophy Foundation, Andy, Tanya, everyone who does such incredible work and has been just such wonderful partners to us in the process. So my colleague Belle is sharing the slide. So Belle, I'll ask you to go to the next slide, please. Perfect. So just wanted to share some important disclosures here, but the biggest thing I wanted to highlight for everyone joining us today is that we are going to be talking to you about an investigational treatment. So this has not yet been approved by the FDA or any other regulatory agency. It's currently in clinical trials, and we're going to talk to you more about exactly where we are in that process. Next slide, please. So before I hand it off to my colleagues, I just wanted to share a little bit more about Sanofi. So we are quite large. There's 86,000 Sanofians worldwide, and we operate in 70 countries. And then next slide, please, Belle. And we have a long history in developing treatments for rare diseases that began over 40 years ago at a biotechnology company called Genzyme, and then Sanofi merged together with Genzyme. So we have a rare disease portfolio that has a global reach with 100 countries with patients on our approved therapies. And then obviously we continue to work diligently to try to bring new treatments to the rare disease community. As Andy mentioned, I'm a rare disease patient myself, so I feel really passionate about this work and proud to be at an organization that really is very committed to the rare disease space. Next slide, please, Belle. And so, to that point, we take this very seriously. So, in 2023, we launched the Sanofi Patient Community Promise, and that's a set of commitments and measurements that we've developed with input from over 80 patient and caregiver organizations worldwide, and it's endorsed by the highest level of leadership at Sanofi. And it's a promise that's organized across the life cycle of a product. So from the very early stages of research through something hopefully getting onto the market. And it includes addressing gaps in the broader healthcare ecosystem with respect to more deeply embedding the voices of the patients and the care partners that we're trying to serve. And we hold ourselves accountable by publishing a report each year on our progress. Next slide, please. And so before I hand it off to my scientific colleagues, really just wanted to say a huge thank you to the DM1 community and to MDF in particular for the chance to engage and to learn. We've been doing this for a couple years now. We were able to hold last year a virtual Sanofi Community Engagement Day, where we heard stories from people living with DM1 and their care partners. And we had over 100 of our Sanofi colleagues who work on this program and also outside of it who are really eager to just better understand the experiences of the community. We've also held multiple smaller advisory sessions over the years where we've had the opportunity to gather feedback on one, just the lived experience of people with DM1 and also those that are supporting them. We were able to get input on the design of the study that we're going to show you today and make adjustments based on that feedback. And also to understand what can we do to help the community understand gene therapy and really kind of grasp what we're talking about. So you'll see here some of the Sanofi team had the privilege of attending the annual meeting that took place in May. And we're just really grateful for the collaboration and the transparency, how open people have been in sharing their stories. So I'm going to hand it off now to my colleague Didier, who's going to dive into a little bit more about what gene therapy is.
Thank you, Tara. Go to the next slide, Belle, please. So it's probably worth coming back to the basics, the building blocks inside your cells. You've heard these acronyms many times. The blocks inside your cells, they carry the information for the cells to function properly. that there are three families of molecules: DNA, RNA, and proteins. DNA, think of it as a big menu in a restaurant. RNA is the dish you order, so it is still an instruction and not the dish itself. And the protein is the dish that you actually get. DNA is therefore the instruction manual. The RNA is like a copy of an extract with the information you need, and the protein is what you get. Next slide, please. So gene therapy is a therapeutic approach in case something goes wrong with the DNA or the RNA. Basically, what leads to how a cell functions properly. It is a one-time treatment. It delivers genetic material, such as a working gene, for example, to change how a protein is produced by the cell. DNA or RNA do not know where to go in the body once they are injected. They need a carrier. We call it a vector that knows how to circulate in the body and target specific cells. The genetic material is delivered to the cells using a vector. Vector, think of it as a package with an address label, and inside is a specific message for the cells. Typically, the capsid of viruses are used as vectors because they have evolved, the viruses, to be very good at getting into the cells. Only the shell of the virus is used, not the inside of the virus that can be harmful for you. Next slide, please. So here is an example using the capsid coming from an adenovirus. Adenoviruses, you and I have plenty in the nose, and that can give you common cold, for example. But the adenovirus knows exactly where to go. It knows how to enter into the cells and deliver its payload. In that case, it's the DNA that goes into the nucleus. In our case, we use capsids from a smaller virus that you see on the right. It's called the adeno-associated virus. It's a small virus, but is associated with no human disease, no known human disease. Once again, we are not using viruses, only the shell that some viruses use. Its use cannot lead to an infection, as the vector doesn't multiply into your cells as complete viruses would. Next slide, please. So myotonic dystrophy 1 is, as you probably know, is called by toxic accumulation of DMPK mRNA. What does it mean? The problem here is the presence of long sequences of CTGs, three of the coding bases present in DNA, in a gene called DMPK. It's what we call it DMPK mRNA. This leads to these long mRNA, as Andy explained to you earlier, that carry these long sequences and are sticky, and they change the way certain proteins work in the cell. Either good proteins may become trapped in the cell, or toxic proteins may be released into the body. Next slide, please. So the buildup of these sticky molecules is called DMPK mRNA accumulation. The CTG repeats create these long DMPK mRNA buildup in the cells. Here, these proteins are trapped and cannot work. Others, more toxic proteins, are also released and explain some of the changes associated with DM1. We are exploring if these can be addressed by a gene therapy approach. Our idea is to degrade these long mRNAs by injecting a therapy that will interfere with those long mRNA using an adeno-associated virus capsid and a piece of DNA. And this piece binds to the DMPK mRNA and have it degraded by the cells. Pablo is going to explain you how that works. Next slide, please.
Okay, thank you so much, Didier. Thank you, Andy, for your kind introduction. Thank you so much, MDF, for the opportunity. Next slide, please. Well, as Andy mentioned in the introduction, Sanofi is conducting the phase 1/2 study, and this is the first gene therapy study for DM1. But before initiating our first in-human clinical study, our team dedicated over 4 years to rigorous preclinical testing. This comprehensive process included in vivo, but in vitro and in vivo in 2 species experiment in order to demonstrate the safety and efficacy of the program. So the program got to the IND clear in November 24, almost 1 year ago. But also the program got to the Orphan Drug designation in Europe, the Orphan Drug designation in the United States, the Fast Track designation, and Rare Disease Pediatric designation also in the United States. And this is important because demonstrate the robustness of the program, because all this designation has been supported by the preclinical study. Next slide, please. Didier and also Andy explained very well the physiopathology of the disease, and also Andy mentioned and Didier the importance to knock down or reduce the DMPKs as the target of the disease. So our targeting is the DMPK RNA. And we are using the transporter that Didier mentioned before also. This is adeno-associated virus. The transporter adeno-associated virus, we call them as the SAN11. SAN11 is our transporter and is adeno-associated vector. Again, it's not a viral, it's adeno-associated vector, it's a recombinant adeno-associated vector. In the cargo that we are using is a microRNA, and the microRNA reduce the level of the DMPK RNA and reduce also the RNA toxic, including the FOCI. The result of this reduction of the DMPK and the RNA FOCI, etc., improve the muscle splicing and correct and leading the correction of the myotonia. And this is the objectives that we have at the end of the day. Again, DMPK as a target, using microRNA, reduce DMPK, improve the splicing, improve myotonia. Next slide, please. And the next slide is a resume that mentioned before, but you can see more clearly the SAR 446268. This is the name of the program, and you can see in there the adeno-associated virus, the cargo, the target in mRNA, and the degradation of mRNA. So this approach offers the precise and efficient method to reduce the DMPK expression and potentially addressing the cause of the myotonia dystrophy. Next slide. Tara introduced Sanofi to you before, but let me introduce to you the Genomics Medicine Unit. 5 years ago, Sanofi established the Genomics Medicine Unit, GMU, in Walton, Massachusetts. And there, around 250 people work only on gene therapy programs. It's not only for rare disease, include gene therapy in NOFTA, include in oncology, inflammatory, cancer, etc. So in GMU, we have the advanced tool like the adeno-associated vector, but another no-virus vector or no-virus platform. Also, we have a program to delivering, as mentioned before, RNA. This is our program. Antibodies, we can replace this missing protein, and we can correct some faulty genes using the gene editing technology. Meaning that all this team and research and CMC and clinicians are fully dedicated to develop the gene therapy program for rare disease, but for another too many indications. I will move on to Belle. Thank you so much.
Thank you, Pablo. Thank you, Didier, and thank you to the MDF for welcoming us today. So now that we've reviewed some key gene therapy concepts and reviewed our mechanism of action, our specific gene therapy, let me introduce you to our clinical study. So what is the BrAAve study? So the BrAAve study, we just recently launched the BrAAve study, and this is a combined phase 1 and 2 study. So as mentioned by Pablo, phase 1 and phase 2 are parts of the process to getting a drug approved or treatment approved. And so this phase 1 and 2 combined study that we have looks at the safety, so basically looking at any side effects, as well as how well the treatment works, or what we call efficacy. So in order to do that, we split the study into two parts. So we have a part A, which is the goal of which is to really select the most effective dose that is also at the same time the safest dose. This will be done by evaluating 3 different dose levels. So as you can see here, dose 1, 2, and 3, with 3 participants per dose cohort. So there will be a review of the safety and efficacy of the gene therapy in the 3 participants per dose cohort before treating the participants in the next dose cohort. So once all participants have received the gene therapy, another review will be done to choose the safest and most effective dose. And that dose will then be given to another 20 participants in the Part B portion of the study. So all participants from both Part A and Part B will be asked to participate in a separate long-term safety study, which will follow them for an additional 3 years to make sure they remain safe and healthy after treatment. Okay, so what is the purpose of the BrAAve study? The purpose of the BrAAve study, as you have seen from the study design, is really to evaluate the safety of the gene therapy, as well as to assess, how the study drug or how the therapy can potentially impact the progression and worsening of the symptoms of DM1. So who can participate in this study? So the study is open to participants with non-congenital DM1 who are aged 18 to 50, so adults for Part A, or 10 to 50 for Part B. So some adolescents are included in Part B. So participants need to have the genetic test for DM1 with CTG repeats of at least 50. Participants need to have implanted cardiac devices, but this is only for Part A. So Part B participants will not need, or is not limited only to participants with cardiac devices. So participants also need to be ambulatory with or without assistance. And participants cannot have any active infections or any malignancies, so no cancers. So what happens during the study? So the study will last about 2 years, and it involves 3 periods, which includes the screening stage, treatment, and the follow-up period. So during the screening period, the participant will be assessed to make sure that he or she is ready to receive the gene therapy. Treatment will then be given as 1 dose only by infusion, after which the participant will be required to stay for 1 day in the hospital for observation. After treatment, the participant will then roll over into the follow-up period, where safety and efficacy tests will be done at specific timelines for about 2 years. So the BrAAve study is currently open to enrollment, and more details can be found at clinicaltrials.gov. So this concludes our presentation. Thank you so much for your kind attention and participation today.
Great, fabulous. Thank you so much for trying to convey this complex topic to our audience. So maybe so we have received a lot of questions, and let's break them according to mechanics of clinical trials, mechanisms, right, and things of that nature. And maybe let's start really working out the difference between the clinical trial activities we have seen to date and the difference with a gene therapy approach. So maybe you can highlight that one more time. What is really the differentiating hallmark in terms of what you are engaging today?
Maybe we can start with so what's really peculiar with our approach is the use of vector, which is part coming from a virus. So it looks scary, but once again, this is not a virus. This is a copy of a protein that comes from a virus. It has no infectious power. But the beauty of it is that viruses are way smarter than we are. They know exactly where to go. And some of them have a very good affinity for some of the tissues. And some of these vectors and the DNA, which is inside the vectors, work only in muscles, for example. So some of it is going to go into the liver because the liver is a filter that gets a lot of these vectors. But the vector is going to work, for example, only in the muscle, which is the case with the one that we engineered here.
So in an ideal world, let's assume the drug modality is ultimately working. So a patient would only have to be treated once.
Yes, that's correct. That's it has pros and cons. The pro is that it's for the patients. You come only once for the infusion, and the infusion has been shown with similar therapies that were set up, for example, for hemophilia before, that can last for years. The therapeutic lasts for years. We're not claiming anything here. We're using a technology that would allow that. You come once. The con side of it is that the body will react against these foreign proteins and will develop antibodies. So this means that at this stage of research, you can only be infused once, but we have good elements to say that one infusion is going to be enough.
A lot of attendees had questions about how can they become part of a clinical trial. So maybe you could break down for us where do the trials occur, and how does patient selection occur, and things to that nature.
Thank you, Andy. So with regards to clinical trials, so we have chosen specific sites where the experts in neuromuscular diseases, particularly DM1, have their clinics, right? So that is our main target, so that they, know how to manage patients and know what your needs are. And so we have these sites open. And so in the event that your primary care physician, for example, you're talking to your primary care physician or your neurologist, and you would like to learn more about the study, you can have your physician reach out to these sites or to reach out to these investigators at these sites, and they can have a conversation to see if you could be a good participant or you can be eligible for the study.
Super. So the next questions are similar with regards to questions about trial participation. So the first question is, hypothetically, if a patient is currently enrolled in another trial, would they be eligible for this trial as well?
So the protocol has specific exclusion criteria. So just to make sure, again, the goal is to make sure participants are safe. So if there is another gene therapy, perhaps, that they're involved in, they cannot participate. But if there's some other modalities, so they may be able to. Again, this is a conversation that needs to happen between their neurologist and the investigator. If their neurologist happens to be an investigator, so even better. So that is a conversation that needs to happen between them to, again, assess eligibility. And the PIs are also in contact with us. So we have that conversation with them as well to determine if, the participant, you're able to safely participate in the study.
Great. So the announced second question here comes in, and somebody paid really close attention to your presentation, which is really great news. So the question is, why in your part A, why do study participants need to have a cardiac device?
Yes. So, as mentioned previously by Didier, the viral capsid and vector is specific to the muscle. And so the heart is a muscle. And so we are being very extra careful in the sense that, a lot of DM1 patients have ongoing cardiac issues, right? And so given that the vector will go and the treatment will go to the heart, we want to make sure that, the heart itself already has some extra surveillance system in place to help deal with any, untoward incidents that may happen. So again, this is a phase 1 and 2 study. And so this is the first time that the therapy will be given to humans. And so while our preclinical data has shown very good safety profile, we just want to make sure that we have an additional layer of surveillance. And aside from this, we closely follow participants using cardiac markers, EKGs, all these other modalities. Again, the goal is to make sure they are safely participating. So just another extra layer of safety surveillance.
Great. And another question that fits perfectly here. So here the attendee would like to know, why is the length of this study so much longer than in other studies we heard previously? And I think here probably your safety focus will make a very nice transition.
Yeah, so one thing to note that the FDA requires 5 years of follow-up for a AAV gene therapy. So that is a regulatory requirement. At the same time, it's good to follow because, again, this is a one-time therapy. We want to make sure, whatever safety that you are safety concerns or if you're doing well in the study, we want to make sure that is continuing. Right. And we want to also see if there's a durability of the therapy and the therapy is still exerting the effects that it's supposed to. So it's really the intention is good to make sure that things are going as they should be going. So that's the main reason why it's lengthy.
So the next series of questions, maybe let's group them together and let's maybe think about in terms of your intended outcomes and functional improvement. So when you thought about the trial, right, and developing the strategy, how do you think about symptom development, symptom stabilization versus symptom reversal?
I can take this. So the mechanism of action meaning that the reduction of the DMPK, our intention is that some of the participant acts is a muscle back. This isn't the final objective, right? That the muscle back. This is that we are looking for. Obviously, we can't tell today is this possible or not because we have to do the clinical trials and evaluate the patient in the different PROs, and this is another measure that we plan to do in order to evaluate how much improve the quality of life of the patient after the treatment. Our intention, based on the different measures that we plan to include, the 10-meter walk, hand open times, hand grip, etc., is demonstrate that the muscle back. How much the muscle back? We don't know yet. So we have to treat the patient to do the follow-up of them, and evaluate after at least 1 year of the follow-up.
Great, thank you. And just to be responsive to the audience, so you referred to PRO. So these are patient-reported outcomes. So the next question. So how do you? So you say you are using this delivery mechanism to improve and possibly reverse muscle impact. How do you think about this? What is known about the delivery system in, for example, crossing the blood-brain barrier and targeting symptoms in other organs and tissues?
So the biodistribution, first of all, these capsids don't cross the BBB. So this is one of the reasons that we don't include congenital population. We include only non-congenital population. So how much will improve the neurocognitive issues? Well, this is an interesting question. So we believe, because some of the previous investigation demonstrate that is we improve the myotonium, meaning is we can't get the muscle back, many of the neurocognitive issues associated to that could be resolved, such anxiety, depression, etc. But for crystal clear, the capsid doesn't cross the BBB, so don't cross to the brain. So this is important to mention. In terms of the biodistribution, we don't have the chance to show all the results. But when you look at the biodistribution, that is muscle preference, I want to say. It's not only muscle, but when you look at the distribution, muscle compared to liver is significantly lower in liver compared to muscle, meaning the mostly of the adeno-associated vector that we infuse go to the muscle, all of the muscle, including the cardiac, as well mentioned.
Great, thank you, Pablo. So just to clarify and to clean up, you used acronyms. So that was the question was right. So the vector will not cross the blood-brain barrier.
Correct.
So
right. Sorry.
Just to make this.
Use the acronyms. I'm sorry.
Yeah. So the next. So let's wrap up with endpoints one more time. The endpoints of your study. So our audience really has clarity with regards to the main primary endpoints of your study. If you could summarize those one more time, that would be great, because we had several questions regarding this topic.
Yes, absolutely. So the primary endpoint of a study is safety, right? This is a phase 1 and 2. So safety is paramount, right? Treatment emergent adverse event is what we call it. So safety profile, we want to make sure patients are safe. But at the same time, we're looking at efficacy. And so when we say efficacy, we're looking at the level of knockdown of the DMPK mRNA. So you want to see how well the gene therapy knocks that down. At the same time, looking at the functional measures. So looking at video hand opening time, 10-meter walk run test, hand grip. So those are your functional measures. Additional things that we will look at in an exploratory level, we're looking at quality of life measures. So Pablo mentioned PRO, so the patient-reported outcome measures. So we're looking at that. What is the impact to your quality of life? What is the impact to your work, your ability to go to work? What is the impact to your sleep? What is the impact to fatigue? So those are the kinds of things that we want to also see, as well as, someone also mentioned death. So the mortality, morbidity rate, so these hospitalization rates, so these are all additional things that we want to look at in addition to just really looking at that biomarker or that level of knockdown.
Great, thank you. That was excellent. So the next question is, and I assume, even though I shouldn't assume, it's around safety, especially in light of recent deaths that occurred in the Duchenne community. What could you tell our audience? What measures do you have in place, and how do you ensure that this is really that this is, while exploratory in nature, it's the protocol is really focused on absolute patient safety?
So the evaluation of the safety in the study is very extensive. So even include a very solid safety plan for prevent particularly the liver toxicity, the cardiotoxicity, etc. So the study included the immunosuppressor therapy with methylprednisolone in the first 2 days in the infusion, the 2 infusions, and then continue with corticosteroid, for mention more generic, treatment by oral route for approximately 8 weeks of the treatment. Meaning that we plan to cover all the potential toxicities related to cardiac, muscle, thrombocytopenia, and others as well. So the protocol burden is very intense in the first 3 weeks, in the first 3 months, because we will ask the patient to back to the site for evaluation, in particular for liver evaluation that is happen 3 times per week in the first 12 weeks. It's important to mention, but obviously the safety aspect is so important for us, and safety first, right? And this is our philosophy, obviously, in particular for the phase 1/2 study. In terms of the recent communication concerning the Duchenne study, etc., some clarification, I think is important. The capsid that we're using is not the same that the Duchenne capsid used. It's completely different capsid. It's the same family, but it's different. The expression of our capsid is much better than another capsid used in Duchenne. And in particular, our preclinical data is absolutely clean. Don't have any inflammatory expression in the tissues. And the doses that we tested in the preclinical is 6 times higher than the doses that we plan to use in the clinical study, meaning that we have the very extensive safety margin compared to preclinical versus clinical study.
And Andy, I'll just add, this is why we really encourage people to have conversations with their doctor or the site investigator so that they really feel informed about this. Because as we said, it's investigational. We have all of this information that Pablo was talking about, but there still are things that we're learning. And so we really encourage people, whether it's a trial like this or really anytime you participate in research, ask a lot of questions of your doctor and your investigator so that you feel like you have an understanding of, what information we know and what that means. And you can discuss that with your family and really make the best choice for yourself or your loved one.
Great. Let me follow up immediately with a question. It's kind of a personal question from an attendee. So they are really excited about this study. They would like to participate. They don't have a good doctor in their region. What would you recommend to them to get more information and to explore the potential of participating?
So the official communication is reflected in clinicaltrial.gov. So, and this link, the patient have the access to the public information, and in there they can find the site where the study is running today. And also they can evaluate the eligibility criteria, the objectives of the study, the duration, etc. So I recommend the people to look at clinicaltrial.gov. This is official information, and I believe they can find the very comprehensive information about the clinical trial, the site, etc.
Mm-hmm. And I'll add, Andy, that this is an early study, so we have sites in the U.S. We don't have so many sites in the U.S., but for people who are interested and able, we do have support to travel to be part of the study. So it's something to think about because they should have an understanding of the schedule because, as Pablo said, there are, different visits. But if someone was interested and willing and wanted to travel, there is support for that, even if there's not a site right in your neighborhood. But clinicaltrials.gov will have that information there. So that's a great starting point.
Great, and then reaching out to the through your physician to the respective site to get individualized answers. And again, this is not a forum here for us to provide individual and responses. This is for general education. So it doesn't happen very often that we get an entire research team. So now I have so now there are two scientific questions, and the first one is kind of related to the vector, so the delivery mechanism of the cargo. And here the attendee would like to know what's the state of knowledge of having these delivery systems that target beyond muscle. So, for example, delivery systems that target the brain or the GI system, or what can you share, like in a 2-minute summary?
So the delivery system, as you call it, is an intravenous infusion. So it's a bag with 100 ml or whatever, depending on the dose, of course. So it's an intravenous infusion, which means that the vector is going to circulate in your body and enter into any tissue that suits it. So it's mostly muscle, as Pablo said. It goes in the liver also because that's a natural function of the liver. It goes almost everywhere, except that it doesn't cross the blood-brain barrier. That's known for that. After that, the body has a way of getting rid of all these foreign proteins, and it's absolutely natural. It's like a vaccine, or it's like after an infection that you're developing your immune system to get rid of it. But it's right now, there are now other delivery systems that are real delivery systems to deliver genes into the brain or into the muscles or into the subcutaneous space or into different places. But it's right now, the field of gene therapy is mostly about, for AAV, at least for adeno-associated virus-based therapies, it's mostly about intravenous, because we are using this incredible ability of these little viruses to go where we want them to go, muscles, liver, or others.
Great. And then the last question, the last scientific question we have here is the difference between this strategy and a CRISPR-mediated strategy. Again, maybe very high level, right?
Sure. CRISPR is the design of the CRISPR is to change permanently a little piece of your DNA. So really the source, the big menu that I was referring to. It's cutting some of this DNA and it's either replacing it with something else or it's sticking back the DNA. So it's very different. What we want to do is just have this little piece where we make this RNA that's going to go into the DMPK mRNA. So the mechanism of action is extremely different. We are not expecting to have something final that lasts forever, as CRISPR would do, because it's editing. CRISPR is once again editing the DNA. This is not what we need.
So thank you. So how likely? So I'm going back to another delivery question. So how do you handle for your trial, and then later, assuming the drug will be approved, how do you handle potential immune responses to the therapy and its associated effectiveness? Maybe you can give us some brief ideas about such management structures.
So it's once again, it's natural for the body to try to get rid of the vector. It's the role of the immune system. It's made for that. So in addition to that, what we want to do is the body to have a normal immune reaction against it eventually, but while letting the DNA and RNA do the job that we're asking them to do. What we don't want to do is to have the body overreact or underreact or having an inappropriate response to this therapy, basically. So that's why we're having this extremely severe, intense safety plan on pinging the immune system twice a week to know how it's going. You know, the white cells and then the liver enzymes and the muscular enzymes, and so all these objective clinical signs that we have outside. That's why the safety plan is made for, to calibrate between an underreaction and an overreaction and to be ready for whatever else can happen.
Great. Okay, so now we always have to end with this, kind of. Let's assume everything goes according to plan. How soon would such medicines be available for the larger community? So if you could just speculate, right? Trial phases ending, and then the drugs coming to all of us.
Well, everything is fine and we can't cross the line as we wish. The intention of our company is that the patient have the access to the treatment, and obviously the plan is to work with the payers to reimburse the treatment for the patient. So this is the plan that we have. Actually, our market access plan already started for initiate the discussion with payer in the future, and we believe at the moment to launch the products, the payer will be available for reimburse the pricing of the treatment for the patient.
Great. And to all of you in the audience who answered this question, right? This is a moving target, and of course we will do everything to ensure, right, that the community is up to date. Whoops! And I just realized I use another jargon. A payer is and somebody like an entity like a health insurance company that pays the bills for when we visit a physician, right, or when we get an immunization, or when we get a treatment.
Yeah, Andy, I think that's it's something that I didn't know until I was a patient, and I appreciate the question that was asked, because I know, someone has waiting for treatments for my disease myself. It's never fast enough, right? But there is this important element. I think a lot of us understand that a regulatory body would give approval, like the FDA at the end of the phase 3 would give approval, but there is that additional step of, making sure that the treatment then is paid for and can get in the hands of patients. And I think the biggest thing that we can say now, because it is early and it's a long process, is just that with all that we've learned and heard from the community over the recent years, we really do have a deep understanding of the unmet need. And so we definitely on the Sanofi side are working with all the urgency that we can to try to move things forward.
Great. And the last cost-related question, of course, it's as you in the audience heard how long such a trial lasts, right? It's entirely too premature to even speculate about costs of such an event. To be respectful of time and to really clean this up one more time, because there are still questions trickling in. The main difference, and here I would like confirmation from you all. The main difference in principle, between a gene therapy approach and the other approaches today is that it is a one-time treatment.
That's absolutely correct. I've seen the follow-up questions coming that's of how long it will last, the effect will last. It's an excellent question. All what we know now is what close but different, like Zolgensma and others have been treated 5 years ago or so, and they're still completely okay. In another field, on rare coagulation disorders, some patients have been treated for 7-10 years ago now, and they are okay. So we are only hoping that the therapy that we're using will follow the same profile and that the therapeutic effect will last for years with an S. We don't know how long that will last.
Great. I think that was a really nice summary, and I think that would be actually a very good conclusion of this very excellent question and answer session. And I see here, right, from how many questions we've gotten, right, and the excellence of the questions. I think we might want to think about doing something like this again, right, in the not so distant future. So with this, I would really like to extend a very, very sincere thank you to you all, our Sanofi friends, right? Please come back. Please keep us abreast what's going on. And then to you all in the audience, if today's program did speak to you, please consider showing your appreciation with a donation to MDF. Follow the link. And also something that's equally important to all of us, if you are living with the disease, please consider participating in the registry. Only if we know about your individual, how you are individually affected by the disease, as a whole, we can manage the drug development process and assist bringing you meaningful drugs. And with this, I would like to end. Thank you so much again, and until soon.
Thank you
much.
Thank you so much for having us.